Hi, this is a video to walk you through how to collect your data on cBioPortal if you're following template research question number 2. So this is the template research question where you can divide up patient data by tumor grade and then look at the impact on genetic alterations, which is one of the pieces of data you'll collect, and then also on patient survival. So starting off on the cBio landing page, the first thing you're going to do is select which of the many patient data sets you're going to analyze. So one of the easiest way to do that is you can filter patient data sets by cancer type. So for this example, I'm going to look at a kidney cancer. So let me find kidney. There it is. And then I am taken to the list of patient data sets that are related to kidney cancers. So there are many different choices. For this example, I'm going to look at kidney renal clear cell carcinoma. I'm looking at this data set that is from the 2020 Pan Cancer Atlas. You are welcome to use any of these. I sort of lean towards the 2020 Pan Cancer Atlas just because I know it's a very large study. So it's more likely there'll be enough samples in order to do my statistical analysis. But you are welcome to choose any of these other patient data sets. What's really wonderful about cBioPortal is each one of these data sets will have a link to PubMed, so you can find that original citation and properly cite it when you're presenting your research. So again, I'm selecting the renal clear cell carcinoma from the 2020 Pan Cancer Atlas. So let me select that, and then down here at the bottom, I'm going to click Explore Selected Studies. So when I do this, it takes me to a data dashboard that is just related to the patient data set that I selected. And looking at different tumor grade, going through all of these different windows in your data dashboard, you're going to look for one that says grade. So sometimes, and in this particular case, that particular panel is not going to be seen. So I can actually add it to my data dashboard. So the way I do that is go up here to where it says charts, click on that, and then I can search for grade. So that brings up neoplasma histological grade. And if I click that box, now it's going to be added to my data dashboard. So back to the dashboard, all the way here at the bottom is my newly added grade data window. It's currently in a pie chart, so to change it, I'm going to go to the 3 lines here in the corner and go down to Show Table. So now in my table view, I can select which of those tumor grade samples that I want to compare. So I do see grades 1 through 4 and also an unknown category as well. For this example, I'm going to select grades 2, 3, and 4. The other grades are there, but the numbers are rather small, and it's less likely that cBioPortal will be able to do a statistical analysis. So I'm going to select my 3 grades that I will be investigating. And once I've done that, I go up to the 3 lines in the corner again and select Compare Groups. So now cBioPortal is working its magic and doing all the statistical analysis and building graphs based on those tumor grades that I selected. So by default, it'll take you to this overlap, so you can see if there's any overlap between your experimental groups. There aren't any here. It's unlikely that you would see overlap when you're examining tumor grade. However, if you do, go back to the video for template research question number 1, and I show you how to break up groups that have overlapping samples. But again, in this particular example, we don't have any. So the first thing I'm going to do is get my Kaplan-Meier survival graph. So I'm going to go to this tab here that says survival, click on that, and that brings up my graph. And depending on the size of your data set, cBioPortal will do your statistical analysis, so it'll let you know what your p-value is. So my p-value is also tiny, tiny, so 10 to the negative 10. So there is a statistical difference between the outcomes for patients based on tumor grade. And if I want to download this graph, then I go up here to the download icon, and I can download it in various file formats to get the graph itself. Alternatively, I can go to data and download the raw data if I wanted to build my own graph in a different program. The other thing that cBioPortal will do for you, if your data set is large enough, it will calculate hazard ratios. I don't see it here for this data set, but you might see it in yours. One more trick about survival graphs before we move on, but if you look by default, you will get overall survival. However, cBioPortal does give you other options. So if I wanted to look at disease-specific, progression-free, disease-free, I can click over on the left-hand side to select for those graphs and then download them the same way I would with the overall graph. All right, so that gets you your survival graph. So the next thing is your genetic alterations. So up here at the top, you're going to click on the genomic alterations tab. And what this is going to do is give you the most frequently mutated genes, and it's going to compare them by your different tumor grades. So if I'm happy with the way this graph looks, just like with my survival graphs, I can go to the download icon and download this graph in various file formats. Right now, it's looking at all of the mutations, but as an option, if you wanted to look at something specific, so let's say I just want to look at missense mutations, I can filter that on the left-hand side, and then cBioPortal will generate a graph just looking at that specific type of mutation that I have specified. And just like before, you can go to the download icon and download that in any file format that you choose. All right, so that shows you how to get your survival graphs and your genomic alterations graphs. Have fun playing around on cBioPortal.